Enzymes are protein catalysts, biological molecules that speed up chemical reactions in living organisms. They are essential for life, enabling countless biochemical processes to occur at rates fast enough to sustain cellular functions. Without enzymes, many reactions that are vital for metabolism, digestion, and energy production would proceed too slowly to support life. Enzymes are biological catalysts that accelerate chemical reactions without being consumed in the process. This means that a single enzyme molecule can be used repeatedly to catalyze the same reaction many times. Enzymes work by lowering the activation energy required for reactions to occur, making it easier for substrates to be converted into products. Enzymes possess 4 key properties. First, they are highly specific, meaning each enzyme typically catalyzes only one specific reaction or type of reaction. Second, they are reusable and not consumed during the reaction, allowing them to function repeatedly. Third, they are extremely efficient, lowering activation energy dramatically to speed up reactions. Fourth, most enzymes are proteins with complex three-dimensional structures, and this structure determines their function and specificity. The active site is the catalytic region of an enzyme where the substrate binds and the chemical reaction occurs. It is a specific region with a unique three-dimensional shape that is complementary to the substrate molecule. This relationship is often described as a lock and key mechanism, where the substrate fits into the active site much like a key fits into a lock. The precise shape and chemical environment of the active site determine which substrate can bind. The enzyme catalysis process occurs in 3 main steps. First, the substrate molecule binds to the active site of the enzyme, forming an enzyme-substrate complex. Second, the reaction occurs at the active site, where the substrate is converted into product molecules. Third, the products are released from the active site, and the enzyme returns to its original form, ready to catalyze another reaction. This cycle can repeat thousands or even millions of times. The lock and key model is the traditional explanation of enzyme specificity. In this model, the substrate shape exactly matches the active site shape, much like a key fitting perfectly into a lock. Only the correct substrate can bind to the enzyme's active site because of this precise complementary fit. This model explains why enzymes are so specific and why they only catalyze particular reactions. The Induced Fit Model is a more modern and accurate explanation of enzyme-substrate interaction. Unlike the rigid lock and key model, the Induced Fit Model proposes that the active site is flexible and changes shape slightly when the substrate binds. This dynamic interaction creates an optimal fit between enzyme and substrate, enhancing catalytic efficiency. The conformational change helps position the substrate correctly for the reaction to occur. Several factors influence enzyme activity. Temperature affects enzyme function, with an optimal range that increases activity, while extreme temperatures can denature the enzyme and destroy its structure. pH level is also critical, as each enzyme has an optimal pH at which it functions best. Substrate concentration affects reaction rate, with more substrate increasing the rate until all enzyme active sites are saturated. Finally, enzyme concentration directly impacts reaction speed, as more enzyme molecules mean more active sites available for catalysis. Enzyme inhibitors are molecules that decrease enzyme activity. There are 2 main types. Competitive inhibitors block the active site by mimicking the substrate structure, competing with the actual substrate for binding. This type of inhibition can be overcome by increasing substrate concentration. Non-competitive inhibitors bind to a site other than the active site, causing a change in the enzyme's shape that reduces or eliminates its catalytic activity. This type cannot be overcome by adding more substrate. Let's review the key concepts about enzymes. Enzymes are biological catalysts that dramatically speed up chemical reactions in living systems. Their active sites bind substrates with high specificity, ensuring that the correct reactions occur. Enzyme activity depends on environmental conditions such as temperature, pH, and the concentrations of both substrate and enzyme. Finally, enzymes are reusable molecules that are absolutely essential for life, enabling the thousands of biochemical reactions that occur in cells every second.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.